Hi everyone, Teacher Janet here. In this video, let's try the Johor SPM Trial Paper 3, 2023 for this year, right? It's the Ujian Amali Science. Code is 4551/3, Biology, Kertas 3, right? And the topic involved is the digestion of lipids from chapter 9. So this is the first page of our exam, the trial exam, which was held November 23. The time is 3/4 of an hour. Okay, so that's 45 minutes. And kertas soalan ni adalah dua bahasa, soalan dalam bahasa Melayu mendahului soalan yang sepadan dalam bahasa Inggeris. So it's bilingual. There are two languages here. So information for you, during the first 5 minutes, you are required to check the checklist, read the questions and plan the experiment, but you cannot start writing. Okay, you cannot start doing the experiment. So the suggested time allocation for answering this question is 40 minutes because 5 minutes has been used for checking. Okay, record all your observations and answers in the spaces provided and show your calculation. Normally show your calculation if there's any. And you're allowed to use a scientific calculator. Okay, so the full marks is 15 marks. All right. So here's the checklist for the apparatus and materials. Now, the instructions. You're not allowed to work with the apparatus in the first 5 minutes. This period is used to check the apparatus list, read the question, and plan the experiment, which is to be carried out. Mark tick in the box provided to check the materials and apparatus prepared and supplied. Okay. So number 1, cooking oil. Number 2, sodium carbonate solution, 0.1 m. Not sodium bicarbonate, sodium carbonate solution. Sodium carbonate solution, 0.1 m. Dishwashing liquid, phenolphthalein indicator and dropper, distilled water, lipase enzyme, test tube and stopper, test tube rack, 250 ml beaker, syringe, stopper, stopwatch, label paper and tissue paper, right? So a syringe is sometimes used in place of the small measuring cylinder in order to measure a certain quantity of a fluid or solution. All right, you can use a syringe, but read the readings on the syringe properly, accurately. So sometimes there is no measuring cylinder, and the syringe is used to measure the specific volume of a fluid. Now let's read the procedure. You are required to conduct an experiment to observe lipid digestion in food samples. So the aim of this experiment is to observe lipid digestion in food samples. As you can see here, the procedure is provided for you. Sometimes the procedure is not provided and you may have to design your own experiment, okay, based on the materials or apparatus provided. So here the procedure is given, all right. It's similar to the procedure in the textbook, that is experiment 9.1, right, on the food test. Now, number 1, prepare 2 test tubes and label them as A and B as shown here. Okay, so label the test tubes with a label paper. Then fill each test tube A and B with the following: 2 ml of cooking oil, 1 ml of 0.1 m sodium carbonate solution, and 1 ml of dishwashing liquid. Okay, so both of these test tubes have the same content at the beginning, right? These 3 items. Cooking oil is the lipid used, all right, the substrate. And then sodium carbonate solution is to provide an alkaline condition, pH, about pH 11, right? And then 1 ml of dishwashing liquid to act as an emulsifier, to emulsify the lipids. We'll discuss more of that later on. So you are supposed to close the 2 test tubes with stoppers as shown here, and then shake both test tubes vigorously after adding the last item, which is the dishwashing liquid. And then this is to mix up all the fluids that have been put into the test tubes. So our fixed variable here is the volume of cooking oil, which acts as the substrate for lipase to digest, right? 2 ml of cooking oil is the fixed variable. Next, add 3 drops of phenolphthalein indicator to each test tube and shake the test tube, right? So after you add the phenolphthalein indicator, the color of the mixture will turn to pink color, because phenolphthalein indicator is pink in color in alkaline solution, like in the sodium carbonate solution. Okay, think of the word alkaline, the K here, and pink. So when the condition is alkaline, then the phenolphthalein indicator will turn pink. Okay, especially at a very high pH of, like, say, pH 11. Now, next, add 1 ml of lipase into test tube A, is shown here, and 1 ml of distilled water into test tube B. So this is our manipulated variable. That means the presence of lipase in the test tube. So A has lipase, but B does not. Okay. Then record the change of color of the phenolphthalein indicator after 5 minutes. So the change in the color of the phenolphthalein indicator is the RV, the responding variable, whether there's a change in the color of the phenolphthalein indicator. So which is our precaution? Our precaution is this one. Shake the test tube well, or previously in step number 3, shake the test tube vigorously, okay, to mix up all the components that you have put into the test tubes. That is the precaution. So here again is a picture showing us what we have at the start of the experiment, or at 0 minutes. So both A and B contain the cooking oil, which is the lipid, sodium carbonate solution, which is alkaline, 1 ml of dishwashing liquid, which acts as an emulsifier, then phenolphthalein indicator, and the only difference is the last item. For A, it contains the enzyme lipase, 1 ml of the enzyme lipase, and for B, contains 1 ml of distilled water, right? So both are pink in color at the beginning of the experiment, because both solutions are alkaline. Now, after 5 minutes, let's look at the results. So this experiment should have been done in the lab, okay, during the school term. And we know that for A, the enzyme lipase inside will act on the lipids, hydrolyze it or digest it or break it down to form fatty acids, which neutralizes the sodium carbonate. Okay, acid will neutralize the alkaline, which is sodium carbonate. And after that, it will create an acidic condition in the mixture. So what happens to the phenolphthalein indicator? It will turn from pink to colorless. So think of the word acidic, C, colorless. Okay, right. But what happens for B? What do you think the results are? So because you use distilled water and not the enzyme lipase, there's no digestion of lipids, no fatty acids will form. The sodium carbonate, which is an alkaline solution, is still present. So the alkaline condition is maintained, and the phenolphthalein indicator remains pink. Okay, so that's the difference in the two test tubes, in the color of the two mixtures after 5 minutes. So before we try the questions, let's review experiment 9.1 on lipid digestion in our textbook. Now let's refer back to experiment 9.1 in the textbook. All right, so these are the results of the experiment. Test tube A and B, similar to this trial exam question. All right, A contains 1 ml lipase, B contains 1 ml distilled water. But here our RV is the time taken for phenolphthalein indicator to turn from pink to colorless in minutes. So it was found that for A, the time taken is 8 minutes, whereas for B, the phenolphthalein indicator remains pink even after 20 minutes. So here in our trial exam, we are only waiting for a period of time of 5 minutes. So some students may ask, can the phenolphthalein indicator turn pink by 5 minutes? Whereas here is 8 minutes. Now it depends on the concentration of lipase used. You use a higher concentration of the enzyme lipase, then the time taken will be faster. Okay, so in A, the phenolphthalein indicator can turn from pink to colorless very quickly if the concentration of lipase is higher. Okay, now the reaction in test tube A. So this reaction is similar to what occurs in test tube A in the question here in the trial exam. Okay, lipid plus water. This reaction is catalyzed by lipase, the enzyme lipase. So this will cause the lipid to be converted to fatty acids and glycerol, right? And the explanation is Lipase hydrolyzes lipids into fatty acids and glycerol in test tube A. So these fatty acids then neutralize the alkali sodium carbonate and reduces the pH of the mixture to acidic condition. So this causes phenolphthalein to turn from pink to colorless. Phenolphthalein indicator to turn from pink to colorless. Okay, so memorize this equation, which may be even asked in the exam, the word equation for action of lipase. Whereas for test tube B, as in this experiment that we are discussing, the explanation is the same. Test tube B is a control experiment. It does not contain any enzyme lipase. There's no digestion of lipids and no production of fatty acids. The alkaline sodium carbonate is still present, not neutralized, so the phenolphthalein remains pink in the alkaline solution. So now let's discuss the questions in the trial exam paper. Okay, so this is Johor trial paper for SPM, paper 3. So first question. Construct a table and record the data collected from this experiment in the space below. The following titles should appear on your table. So remember to highlight all this. Test tube, you must have the title test tube, content, and then change of color of the phenolphthalein indicator after 5 minutes. Okay, 3 marks. So the drawing of the table is an important part of the paper 3 or Kertas 3 question. So remember, you must know how to draw the table. Now, first of all, you check and see whether any units are involved. So there's no quantitative measurements here, no measurements here. So there's no units like ml or whatever involved. All right. Now, if that is, you should actually write down beside the title, like volume. Then you should write ml. Okay. First, then when you draw your table, you remember the units. So remember, if the title has any units, you have to write it in. Right. Now let's look at the answer. Before this, can you please try and draw the table yourself? All right. So take out a piece of paper and try and draw the table based on these 3 titles. Test tube, content, change of color of the phenolphthalein indicator after 5 minutes. Okay. So pause the video for a while. All right, let's look at the answer. Okay, here's your table. Test tube, A or B, content. A contains lipase. So content can write down there. Contains lipase or just lipase. Then B contains distilled water. That's the difference between the two of it, right? Now, change of color of the phenolphthalein indicator after 5 minutes. So you have to copy the exact title from the table. Don't suddenly write time taken for a phenolphthalein indicator to change color. That's not the title that they want you to write. Okay, so the title is the change of color of phenolphthalein indicator after 5 minutes. So copy that down into your table. Exactly, copy exactly, right? Now, so what's the change in color? For A, the phenolphthalein indicator changes color from pink to colorless after 5 minutes, correct? But for B, the phenolphthalein indicator does not change color, it remains pink. You must mention what color it is, so don't just say does not change color. You must mention the color. It is pink, it remains pink. Okay, how are the marks given? Now, 1 mark is for the title. You have written test tube, content, change of color of phenolphthalein indicator after 5 minutes. Follow exactly from the question. 1 mark is for the MV, manipulated variable. In this case, the presence of lipase. So A has lipase, B doesn't have lipase, it has distilled water, right? And the third one is the result, is your RV, okay? So the content is your MV, and the change of color of the phenolphthalein indicator after 5 minutes is your RV, your responding variable. So 1 tick for this, if you get the 2 observations correct. So 3 marks. Right, question B1. State the observation for the experiment that you conducted. 1 mark. Okay, so pause the video and try the question first. Now let's go on to the second question. First, state 1 inference from this experiment. Okay, so an inference is what? What is an inference? It's an explanation for your observation. Okay, explanation for observation. Okay, so try to remember what you must have, the components that you must have for observation. Okay, this one, observation, and for inference. And make sure that when you answer the question, you have all the requirements. So pause the video and try the question first. You need to try it yourself first. And then look at the answer afterwards, and we'll discuss it. And if you have made any mistakes, please take note of your mistakes so that you don't repeat it in the exam. Okay, don't repeat it again. All right, so pause the video for a while and try the 2 questions. Right, let's look at the answers. For observation, B1. Now, observation must have what? It must have the MV, RV, and the any measurements or any values, right? Now, in this case, your MV is whether lipase is present or not, presence of lipase. All right, so if you follow the table, you can write in test tube A, where lipase is present, the phenolphthalein indicator changes color from pink to colorless after 5 minutes. Okay, all right, because the RV is the change in color of the phenolphthalein indicator after 5 minutes. Okay, so we can say the phenolphthalein indicator changes color from pink to colorless after 5 minutes. Okay, let's see. Now, your MV is presence of lipase. So here you must write for A, the lipase is present. Okay, and your RV is the observation, which is the phenolphthalein indicator changes color from pink to colorless after 5 minutes. Okay, so there's no measurement or any value to write inside, so we don't have to write that. Now let's look at inference. All right, so how did you fare for your inference? So we said inference is the explanation for the observation. So ask yourself, why is it when in test tube A, when lipase is present, the phenolphthalein indicator changes color from pink to colorless? Why is it? So give the reason. Okay, so in test tube A, the lipase hydrolyzes the lipids in the cooking oil into fatty acids and glycerol. Okay, and then the third point, fatty acids form an acidic solution, causing phenolphthalein indicator to turn from pink to colorless. Okay, so that is your answer. 2 marks. So the first mark will be for the fact that lipase hydrolyzes the lipids, right? And second or third point may be the second mark, right? So here what you have done is that you have stated when there's the presence of the MV lipase, right? How does it make the indicator to change color? So your explanation here is P1, P2, and P3 here, we have mentioned that the RV, we have mentioned the RV again, all right? But we have explained why the RV is like this. Why does the phenolphthalein indicator turn colorless, right? So explanation has been given, okay? And you have stated the MV and the RV in this case. So regarding the observation and the inference, here I have analyzed the answer to help you see the various components, okay, for observation and also for inference. Now let's start with observation. So the answer given was just now in the same slide, in test tube A where lipase is present. Now the MV is the presence of lipase, okay? And then the phenolphthalein indicator changes color. Now that is the sub-RV, all right? And then from pink to colorless is actually the actual observation, which is a qualitative observation. It does not involve any measurements. It's not quantitative, right? So it's a qualitative observation. That means from pink to colorless. So you have MV, sub-RV, and then your observation in this case, all right? Then for inference, we must have the MV, the sub-RV, and also the main RV, and how to link them together in the explanation. Must state the explanation, okay? So that means we are trying to explain this observation. Why does the phenolphthalein indicator change color from pink to colorless? So in test tube A, the lipase, now again this is your MV. What did it do? It hydrolyzes the lipids. Hydrolyzes lipids in the cooking oil, this part of the explanation, into fatty acids and glycerol. So the main component you must state is the fatty acids, which is your main RV. You see, because of the presence of lipase, finally it leads to the presence of fatty acids, which are the products of hydrolysis. So this is your main RV that caused the acidic solution to be formed and then caused the change in the phenolphthalein indicator from pink to colorless. Okay, so mention the main RV, the presence of the fatty acids, and then go on with further explanation. The fatty acids forms an acidic solution causing phenolphthalein indicator to turn from pink to colorless. So this is your sub-RV. phenolphthalein indicator turns, okay, color from pink to colorless. So even you have mentioned it above, so probably you won't get marked for this, but your marks will be on the explanation. MV, RV, and the explanation. Okay, MV main, RV, and explanation. Question C, complete table 1.1 based on the experiment. Okay, so in the table you can see that you are asked to give the manipulated variable and the method to handle the manipulated variable. Okay, so only one variable is asked. And then secondly, question D, state the hypothesis for this experiment. Okay, pause the video and try these two questions first before we discuss the answers. Okay, so practice the trying the question, trying to answer the question. Don't just look at the answers, right? Okay, then at least you will know where your mistakes lie. Right, let's look at the answer. So what is the manipulated variable? Yes, it's the presence of lipase. And what is the method to handle the variable? Now, for manipulated variable, we have to use these terms. Use different or change, all right? So in this case, the answer given is to use the word change. Change the content of the test tube by adding either lipase or distilled water, okay? So change the content of the test tube, and we must mention by either adding lipase enzyme or distilled water. Now, if you're using the word use different, you can still answer the question. Use different solutions, such as lipase enzyme and distilled water, to add to the content of the test tubes. Okay, so use different solutions, and add the word content of the test tubes, because the manipulated variable depends on the, or is related to the content of the test tube. Okay, right. Next, the hypothesis. Now, for this experiment, we cannot use the format as MV increases, RV, MV, RV, sorry, as MV increases, RV increases or decreases. We cannot write that. Okay, so our MV is presence of lipase, which we must mention, and then our RV is the hydrolysis of lipids, which changes the color of the phenolphthalein indicator from pink to colorless, right? Okay, let's look at our hypothesis. Lipase, okay, that means the presence of lipase, hydrolyzes lipids into fatty acids and glycerol. Now, this is actually our RV, all right, the presence of the fatty acids and glycerol, especially fatty acids, which causes the phenolphthalein indicator to change from colorless, from pink to colorless, right? So, but this is not mentioned here. You can also mention. So our main RV is the presence of the fatty acids, but our sub-RV is the change in the color of the phenolphthalein indicator from pink to colorless, okay, which can be added in. So MV is the presence of lipase. Also, we mentioned lipase, and then hydrolyzes lipids into fatty acids and glycerol. So if you're not sure what to write for the hypothesis, always go back to the objective of the experiment, which is stated at the beginning of the procedure, at the beginning of the question. And you refer back, the question says that the objective is to study or investigate lipid digestion, okay, by lipid digestion by the enzyme lipase. Okay, so that means you have to ask the question, what is lipid digestion by lipase? Okay, so the lipid digestion means involves lipase hydrolyzing the lipids into fatty acids and glycerol. Okay, so lipase is your MV, and the RV is fatty acids, and also sub-RV is which causes the phenolphthalein indicator to change from pink to colorless. Okay, you can write this down here also. All right, and then the relationship is that lipase hydrolyzes the lipids into the fatty acids. So we must have MV, RV, and relationship, correct relationship for hypothesis. Now the next question, based on the experiment, state the operational definition for hydrolysis of lipid. So operational definition, what are the components of an operational definition? The operational definition is the definition based on the experiment. Okay, so you can try to write the answer. You can pause the video and try to write the answer. And remember the acronym DORAM. Okay, so you can try the question first, and after that we'll discuss. Pause the video for a while. All right, let's look at the answer. So we are going to use this acronym DOREM. D for definition, the basic definition for the term, like hydrolysis of lipid. And then O for organism. If there's any organism involved, please remember to mention it. Then we have the R for RV and A for affected by, M for MV. Okay, so let's look at our answer. Now, always begin with the term that is being asked, the hydrolysis of lipid. Okay, so the hydrolysis of lipid is the process of breakdown or digestion of lipid into fatty acids and glycerol. So we know the word hydrolysis means a process of breakdown involving water, right? In this case, the substrate is the lipid, so it's the breakdown of lipid or lipids into fatty acids and glycerol. Must mention fatty acids because fatty acids is the substance that causes the change in the color of phenolphthalein indicator to make the condition acidic, right? So this is our basic definition. There are no organisms involved, so we don't have to write about the organisms. But remember, try to write the word process. If it is a process, write down the word process. Also important word, a keyword. Now, and it is shown by the change in the color of phenolphthalein indicator. So here we are going to use or state the RV, the change in the color of phenolphthalein indicator. Okay, to put in the RV, we must put in, and it is shown by the RV. It is shown by, okay, so and it's shown by the change in the color of the phenolphthalein indicator, right? Then the last point is affected by MV. So the hydrolysis of lipids is affected by the MV, which is the presence of lipase. Okay, so the components, DOREM, is all in this answer, right? To get 2 marks. Next question. Predict the observation in test tube A if it is put in a water bath at 37 degrees Celsius. So in the original experiment just now, the test tubes were not put into the water bath. Okay, they were left at room temperature. So room temperature is how many degrees Celsius? Around 28 degrees Celsius. Okay, but now in the second experiment here, you are going to put the 2 test tubes in the water bath at 37 degrees Celsius. So what is your observation? Right, try this question first by and then pause the video first and try the question, right? Okay, let's look at the answer. So here the variable, the manipulated variable is no longer the content, whether there's the presence of lipase or not, right? In this case, we are comparing test tube A in the first experiment with test tube A in the second experiment. So the difference is the temperature. Test tube A in the first experiment is at room temperature, about 28 degrees Celsius. Test tube A in this second experiment is at 37 degrees Celsius. So here the MV is changed to, the manipulated variable is actually the temperature. Okay, so then what you need to observe is the difference is not just whether there's a change in color of phenolphthalein indicator. Both will have a change, okay, because both have lipase. Both test tubes A have lipase, but the time is different, okay? So the answer should be the time taken for the phenolphthalein indicator to turn colorless, to turn from pink to colorless, in this second case, will be shorter or faster than 5 minutes, okay? So that means at room temperature, the time taken for the phenolphthalein indicator to turn colorless could be about 5 minutes, right? But in this second case, it is much faster than 5 minutes. Because why? 37 degrees Celsius is the optimum temperature for the reaction of the enzyme lipase, right? That means the enzyme lipase is the most active at 37 degrees Celsius, body temperature. Thus, the rate of reaction of lipase increases as the enzyme lipase is the most active. Okay, so P1 is the prediction. P2 and P3 are the explanations. So here there are actually 3 points, but we can write it all in to make sure we get our 2 marks. In fact, can also write at 37 degrees Celsius, the enzyme lipase is the most active. Enzyme lipase is most active at 37 degrees Celsius. Last question. What is the purpose of adding dishwashing liquid into each test tube? Okay, so you can pause the video for a while and answer this question. What is the purpose of the dishwashing liquid? Right? 2 marks. Okay, let's look at the answer. First point, the dishwashing liquid emulsifies the lipids and breaks them down into tiny droplets of lipids. Okay, that's the meaning of the word emulsify. In order to increase the total surface area of the lipids for faster hydrolysis by the enzyme lipase. Okay, so dishwashing liquid acts like the bile in the small intestine, okay, which is bile which is secreted into the small intestine. It helps to emulsify the lipids or breaks the lipids down into tinier droplets. So this will increase the total surface area of the lipids for faster hydrolysis by the enzyme lipase. Right? So any 2 points here, but write down all the 3 points. It's better to write down all the 3 points. Okay? Right, that's all for this lesson. Thanks for viewing. Please do share, like, subscribe, and you can also comment, okay, below. So till we meet again, goodbye.